Hi guys, we have done a bit in lab already on microbial metabolism. We've been looking at some different selective and differential agar plates, tubes, things like that. And we've done some metabolic assays as well to look for certain enzymes. So here now in lecture, we're kind of catching up with that material and talking a little bit about what it means to study this kind of stuff, all right? So what we're doing is we're continuing to differentiate organisms. We've already looked at how it is we can see macroscopic and microscopic shape characteristics on that level, and we've seen how we can look at structure of, let's say, the cell wall by using stains. But now we're going further, and we're trying to differentiate among these organisms based on what they can do metabolically. Now, first thing would be to kind of come up with a basic definition of metabolism, all right? A lot of times in my anatomy and physiology classes, I'll pose the question as to what is metabolism, and something to the effect of how we break things down is suggested. They look at me, I'm kind of a skinny guy, and they say, well, you have a high metabolism. I can eat stuff and I don't gain weight. Well, this is just one side of metabolism. Metabolism refers to all chemical reactions that occur that maintain life, okay? So it's a sum total of all of the entire energy budget of the cell, right? Whether you're putting energy in to build something up, or whether you're taking energy out when you break it down. So it's really both of those things, not just the set of reactions that break things down. We have a side where we're building things up as well. Anabolic processes, anabolism, would be a case where we build up bigger molecules from small molecules. So here we have an example with the amino acids here. Amino acids would be the building block of proteins, of course. We've learned that with our basic chemistry stuff. And those amino acids are going to be built up into, let's say, a protein. This is anabolism. Think of anabolic steroids maybe, because that will remind you that somebody gets bigger, right? So you're building something bigger. This typically would require energy input. So think of all of these bonds. Think of all of these peptide bonds in this case that are holding these amino acids together as not just electrons that are being shared, right? Because we know that each of these bonds, we could talk about the electrons being shared there in a covalent bond. But you can also think of this as energy that needed to be put into the process and is now harnessed in that bond. And the more bonds we have, the more energy we have in the molecule that's being produced, okay? So this is an endorganic. You're putting in, think endo, right? So you're putting in energy to build up these things during anabolism. Now, on the flip side, we have these other catabolic processes, catabolism, right? So this would be a case where large molecules get broken down into smaller ones. This typically is going to release energy, an exergonic reaction. And here's an example. We have this complex carbohydrate, glycogen. Could just as well have been a starch that we choose here, but we have these different glucose molecules, and they're being held currently together by these bonds, and then they get broken into the monomers, right, into the monosaccharides like glucose molecules here. And that would release energy. All of those electrons would represent energy, and those electrons, wherever they go, they might be used in another bond someplace else, okay, after these are broken. So we've got two distinctly different sides to this metabolic set of processes. We have catabolism, where we've got these subunits that are broken down into individual molecules, and this would liberate a certain amount of energy. And then if we're going to build the molecule back up, we need to put that energy back into the process to make these bonds, which they didn't represent in this image, but there would be bonds basically holding these subunits together, right? Now, where that energy comes from could just be from the the light from the sun. All right, so that is possible that you could get the energy in that way, or you could break bonds in a molecule and steal that energy, right, to make bonds in another place. Now, when we consider where this energy is coming from, right, the energy source, we could think about those particular organisms that harness the sun's energy in the form of light, okay? And we'd put photo in front of a word if it was describing an organism that does that, okay? So if an organism gets its energy to form bonds of its own from the light from the sun, we'd say it is going to be a phototroph. Okay, so it's going to be a phototroph. When it comes to energy that is gleaned from breaking bonds and taking that energy, okay, from the chemical bonds in another molecule, well, organisms that do that we'd say are chemotrophs. Okay, so they're going to break bonds in chemicals and take those bonds and reform them elsewhere, right? Take the electrons from what the bonds they broke and form them in another molecule. So we have chemotrophs and phototrophs. Now, so I'm going to put sun here and sun down here where we have light, right? So light energy from the sun. We have phototrophs. And the classic example of something like that would be a plant, right? So plants, anything that's green that's growing like that, we would say those pigments are helping these organisms to harness that energy and use it in their own bonds for their own molecules. Now, where do we get our energy? Well, we get our chemical bonds energy from the food we eat, right? We're not just soaking up the sun and using that energy to form bonds like a plant can. We have to eat food to get that energy, right? So a chemotroph, like all animals, okay, and some bacteria and other single-celled organisms, microbes in fact, right? A lot of them have to eat food, right? Like us, we have to eat food to have energy, and we break down the bonds in that food and we use that energy to make bonds in our own molecules, okay? Now, if we consider the carbon source, okay, so where are all the carbons coming from? In my lipids, in my carbohydrates, in my proteins. These are all organic molecules, right? So they're stocked full of carbons and some hydrogens and oxygens and other atoms that we know of. But what is the carbon source, okay? Now, if that carbon source is an inorganic compound, we say that the organism is an autotroph, okay? If the carbon source is from organic compounds, we'd say that this is a heterotroph. So, if we consider an animal like ourselves, right? Well, not only do we get energy by breaking down the chemical bonds in the food we eat, but the food we eat also provides us with all of the carbons, all of the raw material for making our own molecules, right? So we eat food for two reasons. We eat food for the energy we get from chemical bonds in that food, and we eat food for the raw materials that it gives us. All of those carbon-containing molecules give us the raw materials for making our own molecules, right? So we would be considered a chemoheterotroph. This is not necessarily altogether different than what a lot of bacteria do. So if we look at bacteria, like those that we've been growing in our lab, is it any different? We haven't talked about putting bacteria outside in the sun to get them to grow. We had to put them in a broth or an agar plate that had certain kinds of nutrients, right? So yeah, they need to eat too, right? So all of the bacteria we've talked about really so far in the laboratories thus far, they all needed some kind of medium that had food. And they're doing the same things we are. A lot of those that we're working with are chemoheterotrophs, okay? They're getting energy from the bonds they break in those molecules, and they're getting the raw materials from the carbons in those molecules. All right, so you want to have a basic understanding of how these roots are used, how these prefixes are used in these terms. And I should be able to give you an example, and you would then use those examples to tell me, is that organism a photoautotroph, like a plant, right? A photoautotroph would be getting its energy from the sun and taking inorganic compounds to get their carbon. In this case, CO2 would be a good example of that. CO2, even though it has a C in there for carbon, this is one of our exceptions. Carbon dioxide, because it doesn't have hydrogen and oxygen together in that molecule, we consider carbon dioxide inorganic. It's one of those exceptions, right? So our plants are photoautotrophs, right? All right, so what we're really talking about here as we get going in metabolism is we need to break molecules down, break bonds, and when we do that, energy is released. And then we can use energy to build and make new bonds. And in the end, these small raw materials that we take in are going to become bigger and bigger and eventually get combined to make what we know as a cell, right? And we grow, right? So this metabolic ability we have to yield energy and then build these things up through an anabolic process, right, will ultimately tell us, can that organism grow or not, and where do they grow, right? Do they have the metabolic capabilities of using what molecules are available to grow, right? Okay, we're going to leave it at that for this particular segment, and we'll jump into enzymes and what they do next.